Let us return, however, to the genetic code, which is important from the point of view of determining similarities between organisms, which is the purpose of this presentation. We remember that the genetic code is used to encode 20 possible types of building blocks from which a protein is composed, that is, 20 different amino acids. We also remember that each amino acid has its assigned codon, or rather several codons, which are encoded in DNA across 3 positions, at each of which one of 4 bases may occur. This means that we could encode 4 times 4 times 4, that is 64 different amino acids, and thus in our example the codons TTA, AGT and ACG will encode a protein containing leucine, serine and threonine. Because proteins are composed of only 20 different types of amino acids, we can therefore expect that a single amino acid will be encoded using more than one codon. A listing of all 64 codons together with the ranges of amino acid encoding can be read from a table in the format most commonly used to present the genetic code. And so, for example, in order to read which amino acid is encoded by the codon ACT, we find the first letter of the code on the left side of the table, which is A. Next, we look for the second letter of the code at the top of the table, which is C, and the third letter of the right side of the table, which is T. Next, we locate the code in the table at the intersection of these letters, ACT, and read the amino acid assigned to it. Here it is threonine. As we can see, similar to the case of the bases A, G, C, T, each of the 20 amino acids has its own name and letter designation. As we can see, the amino acid threonine is encoded using 4 codons: ACT, ACC, ACA and ACG. That is, when one of these 4 codons appear in the DNA, this particular amino acid will appear in the protein. In general, some of the 20 amino acids are encoded by 2 codons, others by 4, and for example, leucine is even encoded by 6. Because DNA is a long molecule containing the coding for many different proteins, there must be a codon that specifies the beginning and end of the protein code. These are the start and stop codons. The segment of the code from the start codon to the stop codon is called a reading frame. It is also worth mentioning here that, in simplified terms, the sequence encoding a single protein is called a gene. As I mentioned earlier, the start codon usually serves as a codon that specifies one of the amino acids in the subsequent part of the reading frame. It is the codon for the amino acid methionine. So to define the end of the reading frame, we need a codon that signals the termination of translation, that is, a stop codon. In human DNA, there are 4 stop codons, which can be seen in the table. These are TAA, TAG, and AGA and AGG. There are some minor differences in the coding of nuclear and mitochondrial DNA. Because we will be focusing on finding similarities based on mitochondrial DNA, therefore the table presented refers specifically to the mitochondrial DNA code. For the sake of clarity, the coding for nuclear DNA is provided in parentheses. For example, in nuclear DNA, the codon ATA encodes isoleucine instead of methionine, and TGA encodes stop instead of tryptophan. We remember that mitochondrial DNA contains 13 genes encoding proteins. For us, however, the most interesting aspect, for the point of view of observing similarities and differences between organisms, is the similarities and differences in the genetic code for these genes. In the genetic code of 2 organisms of different species, for example comparing the mitochondrial DNA genetic code of a mouse and a human, we can observe fairly radical and relatively rare differences in the form of the additions or deletions of entire codons. that is, triplets of coding bases, referred to as insertions and deletions. But most commonly, the differences involve base substitutions at individual positions, just called substitutions. Thus, for example, if a substitution occurred at the first position of the codon CTC, which encodes leucine, changing C to A, we would obtain the codon ATC, which encodes a different amino acid, isoleucine, potentially introducing greater or lesser changes in the structure of the protein. According to the evolutionary model, over a long period of time, a process of continuous random mutations occurs, mainly causing substitutions, that is, base substitutions in DNA, which may have no effect on the structure of the protein. For example, a change at the third position from A to C, that is in the codon for valine, that is from GTA to GTC, will not cause a change in the encoded amino acid or affect the protein structure, as can be seen in the DNA code table. Some substitutions will affect the change of an amino acid and thus the structure and adaptation of the protein's function to the new complexity of the organism and changes in the natural environment. It is assumed that the most of these random mutations will introduce harmful changes, preventing the survival of the organism, or changes that impair the function of the protein, weakening the organism and reducing its chances in competition for food and a genetically strong mate in order to pass on its genes to future generations. Some changes will however, have a positive effect, giving an advantage over other organisms in the struggle for survival and passing on beneficial genes to future generations. It should be mentioned that the bases A, G, C, and T are divided into two groups. Into the larger purines, which are the bases A and G, and the smaller pyrimidines, which include the bases C and T. As a result, base substitutions of similar size, that is within the same group, may occur more frequently than substitutions between these groups. Substitutions within a group, that is between A and G and between T and C, occur more frequently and are called transitions. Substitutions between groups occur less frequently and are called transversions. Transitions and transversions are best illustrated with a specific example. In the comparison of two fragments of the mitochondrial DNA genes from human and mouse, we can see the human DNA codons and their corresponding mouse DNA codons. Just above, we can see the rows showing the individual positions of the codon. We can see the row of transitions, labeled as U, the row of transversions, labeled as V, and above them the row indicating the position at which the substitution occurred. So, let us take the first codon in the row of human mitochondrial DNA codons. We can see the codon CCC, which encodes the amino acid proline designated as P. In the transitions row, we can see in relation to the mouse codon two transitions at positions 1 and 2. We can see that the mouse codon is TTC, which indeed represents a transition, that is a change of the codon from C to T or T to C at two positions. The direction is not known to us. This change was very significant because it led to a difference in the amino acid proline in human DNA and phenylalanine designated as F in mouse DNA. The amino acids for the individual codons are provided in the rows between the codon rows. Between the rows of human and mouse protein amino acids, there is a row showing the alignment of the amino acids. Identical amino acids are marked with black lines. A colon indicates amino acids with similar biochemical properties, that is, ones for which a change does not necessarily introduce radical differences in the protein structure. The absence of a symbol indicates a significant difference in the biochemical properties of the amino acid, which may introduce substantial changes in the protein structure. Moving to the second codon, we can see two transversions at positions 1 and 3, where the codon ATG encoding methionine M is changed to TTT encoding phenylalanine, shown as F. Both amino acids are similar and are connected by a colon. It is worth mentioning that the genetic code is considered to be degenerate, meaning that it has a surplus of possibilities relative to the requirements for encoding amino acids. We have 64 possible codons at our disposal, but only 20 amino acids to encode, plus a stop codon. Thus, there are 64 minus 21, that is 43 redundant coding possibilities.